Okay. So, the first lecture is on molecular graphics. That may seem like something primitive, but I like to make two comments that come really from the bottom of my heart about the role of molecular graphics. The first one is actually, even though it's a little tongue-in-cheek, very serious statement from Yogi Berra, who is cited very often. And I think he said something like, you can see a lot just by looking. And what it basically means is that you just are well advised in any kind of science and in any walks of life to look. I recommend to my students that they almost spend an hour a day to look at their proteins, to look at their trajectories, and not to just be blind about results, be schematic, but just use the human mind, the incredible intelligence that we have in our visual system, and that is what molecular graphics is doing for us. The second comment is that, molecular graphics is a mundane thing, it's just graphics, it's not nothing, deep. And you think that it's like the stupid sister or the stupid brother of modeling. But, that's really crazy because modeling itself, molecular dynamics, is, like you go to this big computer, you are getting a lot of numbers back, but how accurate are these numbers? Many of the things that you're getting out of modeling is also very qualitative. Even though you're dealing with numbers, that doesn't mean it's quantitative. They are ideas. And a little bit is a problem that using the computer today, you're running a little bit out of ideas and you just become too schematic. People, for example, publish many papers nowadays just because they let a protein wiggle and then they think they have the right to publish in Biophysics Journal a long paper on it. If you think about it, then molecular graphics and molecular modeling are really, in a way, the same. They are a very qualitative approach, a systematic but qualitative approach to biology. They are a systematic way of thinking carefully in detail about the system. Rather than having some crazy ideas, you look at it with molecular graphics. And rather than having some crazy ideas, you apply the law for physics to see if it sort of makes sense or if things clash into each other or if the laws of Newton are totally disobeyed or something of the kind. And so in a way, both molecular graphics and molecular modeling are just like what the pen is for the mathematician. Good penmanship, a mathematician will tell you, is actually important. Good notation, good penmanship is maybe half of the secret of being a good mathematician. You find very often that they have good penmanship and they're very neat when they're good mathematicians. And so here we have molecular graphics, molecular modeling being our penmanship. And let's think about it that way. There's really a lot of truth in that. And if you think about it this way, then you will immediately agree molecular modeling, dynamics, very fine, but molecular graphics is also very fine. Both of them should be done. Both of them give us an opportunity to approach our subject carefully and systematically. And so in this regard, molecular graphics is not the poor sister or brother of molecular dynamics, but rather an important intellectual tool, or let's say a tool to give us an opportunity to think intellectually about our system. The molecular graphics program that we will be using is the program that we develop here. It's called VMD. I don't want to sell VMD to you. Like first we don't make money on it, and there are other good graphics programs around. We have many users. I think maybe it's written somewhere. We have, I think today, almost 20,000 people who downloaded VMD, and I think probably we have close to 10,000 people who are actually using it really, as we sort of not speak every day, but they they are doing it. We can judge this quite well from whenever we have when we have a release, we see that many people immediately get the latest version, so they obviously have an interest in it. It's a very beautiful graphics program. It's a very versatile graphics program because of its scripting ability. And so it's sort of like what you may call a computation intelligent program. It doesn't cater to the simplest kind of computing people, but it's also a program that once you know it a little bit, you know how it sort of works, and you can find your way around without having always a fancy manual. So here you see that we have many users. It runs on basically every platform that biologists use, particularly also now on the Macintosh. You see I have it here running on the Macintosh. I'm glad to show it to you. It is very good for displaying large biomolecules and trajectories, particularly trajectories. And it also becomes more of a program that combines sequence analysis and structure analysis. So it started out as a pure structure analysis program, but we have now a sequence window, and in the future we will actually have multiple sequence window and sequence analysis windows. And most important, and we make a lot of effort to teach you this, because it's a little bit harder to learn, it permits extensive scripting that makes it an extremely versatile program. So here you see some of the large systems that this program can handle. For example, I forgot his first name, Dr. Sobamatsu from Los Alamos really recently did a simulation of 2 million atoms of what was it? The ribosome. And he had no problem just getting it all without changing anything in the program, getting it the whole thing into VMD and analyze the trajectory and look at it, rotate it around. So it's really a program that is very flexible in terms of size and also in terms of having trajectories. Now, during the first part of the workshop, we make two friends in terms of doing our analysis and our modeling. And one of them is a very old pet protein of theoretical biologists, namely bovine pancreatic trypsin inhibitor. It's a small protein. It has only a little over 500 atoms. And so, of course, it's wonderful for us to do some simulations. But actually, we also introduced to you another protein that's called ubiquitin. It's, the name comes, of course, from ubiquitous. It's a very common protein, but it's a very modern protein. You found actually several articles on ubiquitin metabolism and signaling pathways in the recent science and nature journals. And actually, all of the uses that we do here with ubiquitin, there's a little hint to you, is really some stuff that hasn't been published yet. And so we are definitely going to extend what we are offering you ourselves in the next few weeks to send our papers out. It's just one of these proteins that really has a lot of beauty and interesting behavior that is very easily accessible, and so we loved it so much when we encountered it as a research object that we felt, my God, why don't we use it for the workshop? Even though we give a few trade secrets away, it's better to work on a modern protein than on the one that is sort of like as old head as bovine pancreatic trypsin inhibitor. So we will actually do mainly our examples during the first 3 days on ubiquitin, and then we give you the opportunity to do BPTI so that you don't publish then and compete with us when you go home, but you might actually also rather work a little bit more on ubiquitin. What is ubiquitin? It's a pretty small protein. Its structure is highly conserved, and it is a tag. It tags itself rather through an evolved system of enzymes to other proteins that are tagged either for destruction, because they are misfolded, or for processing in the cell's nucleus in other parts of the cell. The ubiquitin is actually a very interesting tag. It usually actually involves several ubiquitins. So if you are tagging a protein, you are not tagging it with one, but with several, 4, 6 or more, and they don't go all around the protein, but rather they all go in the little chain on the protein. So there is it's a C-terminus, you have one ubiquitin, and then the other ubiquitins add on. And now there is one little secret. The way they tag themselves on is actually itself a very important message. The ubiquitins have how many lysines? 7 lysines. They are highly conserved. And the chemistry of tagging one ubiquitin to the other ubiquitin works very well by either having them chained terminus to terminus, head to tail, or having them chained terminus to one of these lysines. For example, one of the lysines is lysine 48, and so then you have then one ubiquitin go to the C-terminus of a protein, and then the terminus of with its terminus, and then the next ubiquitin doesn't go to the N-terminus of the ubiquitin, but rather to the lysine 48. And then the next one goes to the lysine 48, and the next one goes to the lysine 48. So they can be head to tail, or 48, 48, 48, or there's another lysine that is also used for this purpose, 63, so they go then 63, 63, and depending on which lysine they go, they form a very straight chain or a very crumbled, more bulky chain. And that can, of course, be recognized by the machinery of the cell. It also looks like, and that's what you will be learning on Wednesday, that these tags that form little chains on the proteins are not just recognition elements, like any good tag in the post office somewhere. It's also a handle, apparently. So like you grab the lysine, the protein by this tag and pull it into the proteasome, where the protein that is tagged this way is being degraded. And that is not completely proven yet, what I just tell you, but it's very likely. And the reason is that there was a dramatic experiment done at Columbia very recently as far, and also not published yet, by the group of Julio Fernandez. He's one of the world experts in atomic force microscopy. And he pulled on all kinds of proteins that exist in the living cells, and he found that those proteins that have a mechanical function, like, for example, the proteins of the muscular system that give muscle elasticity, namely titin, that is made of the longest protein in human body is titin. It has 300 protein domains. They each have particular elastic properties. And these are immunoglobulin domains or fibronectin domains. And so he studied them, and he found that when he tried to stretch those proteins, they are very, very strong. They resist the stretching. But if you stretch too much, then they easily give. So you stretch, and then it takes a while, that suddenly they rupture. So they are designed for mechanical stability and at the same time for large extensibility. The same for fibronectin, that is also part of the titin in the muscle, but it's also part of the extracellular matrix, where the cell adheres itself to other cells or to a substrate, the ground, in order to crawl and move along. So it has to anchor itself with an elastic anchor, and this is done by this protein fibronectin that interacts with one of the most important cell surface receptors, integrin. And if you stretch fibronectin, you also find it's very mechanically stiff, but then at a certain point it gives. There are other proteins, so-called adhesion proteins, for example, in the immune system, that likewise have this kind of property that when you pull on them, they are resisting the pull, but then they give. How about normal proteins, quote, like, let's say, cytochrome or myoglobin? These are usually proteins that when you pull, they immediately give. They have no particular mechanical stability. And now, so you can basically say, it might be a little bit simplified, but in terms of the experience that atomic force microscopy gives us so far, we see that, quote, the normal protein without a mechanical function, when you pull on them, they immediately stretch out, whereas those with a mechanical function are designed for stability. They stretch out also, and apparently designed for stretching out, but you must overcome a certain resistance. Now one found that ubiquitin is exactly of the type of mechanical proteins. You actually have to pull it rather strongly to make it give, but then actually, again, like the other proteins, it stretches out very nicely. And so this makes it likely that the tag that is being used here is actually both a recognition element, a signaling element, designating a protein for a particular destination in the cell, as well as a mechanical element. And that is why we want to study this protein here as an example. It's at the same time very small, and you know, what is very fancy research yesterday is cool, is textbook material today in biology, and sometimes when you are lucky, even, you know, simple example for the student laboratory. Now I mentioned already that the ubiquitin signaling pathway is controlled by certain enzymes. They have to prepare the ubiquitin for this, for the tagging. They have to also recognize which proteins should it be connected to. And these are three famous proteins, E1, E2, E3, and I think I've seen at least in the last half year 10 articles in Science and Nature and PNAS on these enzyme systems. So I mentioned already the ubiquitin tag degrading proteins that are misfolded, and they are tagged for degrading. They also send proteins to certain parts of the cell. For example, in cell division or in gene expression, some proteins are sent with the ubiquitin tag into the nucleus. Here you see a little overview of how the tagging works when the protein goes to the proteasome, where the protein is then pulled into the proteasome and then unfolded, and at the end in the process, the ubiquitin is being taken off as a protein is being processed by the proteasome. It's also involved in gene expression around the nucleosome, and the signaling of the ubiquitin depends on the length of the chain, how they are attached together. I mentioned that there are these lysines, and you can attach them through the lysines, and you find usually that in a polyubiquitin, they're always attached the same way, one to lysine 48, to lysine 48, to lysine 48, and not a funny mixture. It also depends where in the cell it happens, and here you see a little bit of what I mentioned earlier, this type of chain linked to lysine 29, to lysine 48, and to lysine 68, and what the artist here drew a little schematically is that the polyubiquitin chain actually then looks different to the recognition system depending on how they are being assembled together. Now I tell you this because of course, we are doing computational biology for the biology, so I do not want to work on ubiquitin here and not even tell you what it is about. But now I would like to go the methodological route and hit on the modeling program VMD, Virtual Molecular Dynamics, we called it, I think, at the time. And the first thing you need to do with VMD is to load the molecule. And so you have then these, when you are loading VMD itself, you get some windows, and then the windows permit you through this file menu here to load your molecule. And so why don't you do that now? Fatime will show you this, and she loads VMD here on the side, and so you see then the main window, and now she goes to file, and she puts their new molecule, and now she don't go too fast. So she goes here to new molecule. Oops, what is? I think I need another one. So she goes into new molecule. This is just the name of the molecule she wants to do. Then she goes, in this case, to browse, because she knows somewhere the file that she wants to use is on her machine. A very nice way of loading the molecule with VMD is just to type in the PDB code, and then it recognizes VMD. It goes to the network, to the protein data bank, and pulls automatically the PDB. So you don't have to do more. But in case that you want trajectory, something more, you go then to your local disk. So which one do you take then? You take then this ubiquitin PDB. That's from the PDB, except that we added already some hydrogen state to make it look prettier. And so now you need something else. Now you load it. She loads it. And now you will see in a moment that it shows up on the window here. And now VMD permits you to rotate the molecule around if you do that a little bit. And you can also translate it around. And you can also scale it, make it a little bigger. And this is, of course, not a very nice representation. And you can choose other representations by now going to the graphics menu. And so if I go back here, you see then the main menu, the graphics menu. And you go pull this down. And now you're getting another menu that you see here that permits you to put in different kind of rendering styles. And so now, Fatime, you can choose some rendering styles. Like, why don't you choose first van der Waals? Then we see how it sort of looks volumetrically. And then you rotate it around a little. And then you can also, this is actually this is a little better machine, but it works also on the laptop very easily on this size molecule, no problem. Okay, now maybe we take tube. So we see sort of like a little bit more the structure. My favorite is definitely always cartoon, where you see the helices and the beta strands described. You can choose now the radius of the helices. For example, they might be too bulky. Maybe you make it a little smaller. Here, go here to helix radius. And Fatime knows this much better than I, so I just pretend here I'm an expert. And the beta sheet you can make a little thicker. You can, when you want to make a pretty picture, you can improve the resolution and so on. Okay, so you have these different rendering styles, and you can also, as you see already on the top here, you can also color to show particular elements in different colors so that you remember what is what. You can draw some hydrogen bonds in there. We do this later. And you have here this sort of like funny rocking motion to see it 3D. That's also something that VMD provides for you. You just go to some element here in extensions that you will see later. And then you just go to movie, VMD movie, and then you say make a rock movie, and then it looks very nice three-dimensional. It can rotate things around. It makes movies of trajectories and so on. Okay, so you also can do multiple representations. Maybe you do first now a surface. And so now we see a surface. We have several kinds of surfaces, expensive and cheaper ones. And here, Fatime colored it according to amino acid type. This is a water-soluble protein, and so we see that we have polar and charged amino acid on the surface, no hydrophobic amino acids. And now we want to add several representations. We want to see sort of what's inside. And so what Fatime now is doing, if you can do that, is make it transparent. And then add a second presentation. So this is now transparent. And now add a second presentation. So she adds now, maybe put inside, see, now she has here a second line. She puts here maybe a cartoon representation inside. So, okay. And you cannot see it that much. It's, what I would suggest is what we do is what I would suggest is what we do is.
The cartoon is also transparent, I think.
That's a problem. That's a problem. Thank you. There's an expert here already. Okay, so now you see, we wanted to actually, but we forgot to do it. It was my mistake. We wanted to make it more transparent. You can choose the level of transparency. So by going to some material section here, you can make it very nicely. Okay, she does it quickly. So, okay, so material 2, I'll leave it material 2, that's fine. That's fine. Oh, okay. So now she can do there, maybe go to 20, okay, good. And so now you see it more transparent, yes, so you see it very nicely, so you can choose it the way you want it. And now, maybe you show us the lysine groups inside there. So we mentioned that they are the important lysines, and so you can just call them lysines, res name LYS. Okay. And maybe with Van der Waals, yeah, okay, so there you see them in blue, and so here you see where the lysines are, and can you find 48 for us? That's the most used lysine, or maybe you just add another one, lysine 48. Okay, good. Res ID 48, okay. Okay, so, and maybe make that a different color. Okay, so this is at 48. So if you rotate it all around, so this is really the fun. You can really look at it at every part of your protein. And the nice, exciting thing is that these proteins can be very big, and you can still very, very quickly look at any corner of it. So this is what I call really looking carefully and learning from looking. You can imagine, since this one really goes effortlessly, that you can really use VMD for intellectual purpose. So now I have a hard test for Fatime. we want maybe we get rid of some of this, or you just switch it off. Switch some of these views off. And now we want to do the following. We want to take a crystal structure and put it into the window, and then we do an equilibration simulation that you will do later in detail. And we want to go to the last frame of this simulation where the protein wiggled away a little bit, and want to see how far it moved away. So we have now two representations. One is the initial crystal structure, and then we load the trajectory, and we take the last frame of the trajectory and also put it in our window. So let's now see what Fatime is doing here. So we both worked very, very hard with other students the whole night. So it's we're all a little tired here, but you are doing really well. I'm really impressed. So this simulation was done in water. You see the water, but maybe rotate quickly that we see that this is not just a water disk, but it's a real box. Okay, good. No cheating here. Okay, and so now you take just the protein part, and what she will be doing here is she just takes the C alpha atoms of the two proteins, the crystal structure and the simulated protein. Actually, can you show us again the crystal structure? It was somewhere off here. Okay, there you see they are not necessarily together. So maybe we put them both in as loops. okay, and the other one too. And then if you could now write a script that just looks at the C alpha atoms of the two, and so she selected them, maybe she did already, I didn't follow. Ah yeah, so this is a key window that you use. Can you first show us where the window came from? Don't close it, because that could be trouble. But so you go here to extensions, and you go here to this window, and then you're getting there this window where you can write in the scripting. So in the old days, it had to be a complicated Unix type window, but now we have a very comfortable kind of editor almost, nicely a window. By the way, maybe this is a good opportunity. Don't look into this thing now. I introduce you to key VMD programmer. This is, maybe you raise your hand so that I don't blind you. That would be too bad. So this is John. So if you have any question about the ins and outs of VMD, there is John. He's the great hero. He got for, at our last renewal, NIH renewal, he got best possible, how do you call it, grade or whatever you call it, score, 100. I thought it doesn't exist. You had to look in the books, but the site visit committee insisted to give him 100. And so there is John Stone with, and you can ask him anything you want about VMD. Okay, so now let's continue here. So Fatemi writes now the commands that identify the crystal structure, that looks at the C alpha atoms, and that move then the two together. And then they're on top of each other. And so this is just a little transformation. So here she identifies the alpha atoms of one set. This is the other set. And now she will move them then together. So this is the alpha of the other set. Set defines then some kind of macro that identifies a certain property that you can then call as another command. And now she moves these two sets together through an operation M. And then you will see then with the applies and these two proteins are moved together. We have only a little screen here, so we unfortunately have to cover it. But you will see then at the moment they will move on top of each other. Are they already? Yes, there they are. See, isn't that beautiful? And so now you see where the protein moved. You're of course not too surprised at the end. You see it's a pretty stable protein. It's really forgiving. It doesn't poop out on you easily. And now we can actually also do the following. That would take a little longer, so we didn't, Fatima and I decided not to do this. We did you will actually also do this for your, in the afternoon, we did an equilibration run where we let now this molecule rotate. Actually, if I click on this, you probably even see it. Fatima was so kind to make, to make everything into movies here, but no, it doesn't work. So what she did here is she, and you will do this yourself, you do then a simulation run of ubiquitin, and the protein wiggles, and then you are checking out where the protein is more mobile and less mobile. Very famous analysis makes all the time. You see here in the blue parts, for example, this helix here, the protein is very rigid, whereas in the amino acid groups here at the termini, particularly this one here, it's very flexible. And so you can do this very quickly, like you will probably even the first time, you just do it in 5 minutes, and then you have this result visually inspected. You can, of course, also write it. Mobility as a function of sequence identity. Yeah, now we are at the sequence. So this is sort of like the new VMD already existing since half a year, where we do not only look at the structure, but we also look at the sequence. And now, maybe we do this, Fatime, that we open now in this extensions the sequence window. So here we have now here the sequence window, and you will then recognize a pattern like this. What happens here? An error. So what we do is you close it, or maybe you try it again, otherwise you just start it again and errors are there to happen. Okay, so what I would suggest is just close it and open it again. And I mean, probably just close VMD. So I can tell you here what we are going to do. So we have now multiple windows, a structure window and the sequence window, and we see here two columns next to the sequences, and these columns are containing here information on the secondary structure. These blue stuff are the helices, alpha helices, and the yellow stuff are the beta strands, and the other ones are the loops and coiled. And we have here a column, the so-called beta column, where the crystallographers put the beta factors, the B factors, that tells you about the thermal flexibility of all the atoms in the crystal. And you can actually also use it. This is usually given in the protein data bank, and you have these numbers, and you know immediately from the crystallography already whether the protein is flexible or not. But you can also use this beta column for your own purposes, where you put your own analog number that characterizes certain properties of the protein. And now what we can do here is we can now identify very quickly the amino acids that make these beta strands. And so this is what Fatima is now doing. So you can see, you can stripe there, and now you know immediately here the amino acids that are making the beta strand. You see them here. You have here a particular representation that you can choose at will. So those amino acids that are in this segment, you can select, and it's in a selection in VMD, and you can show it in Van der Waals, whatever. And now you can do that for all the other beta strands. And if you do this, you can also use now, maybe you can go quickly to the VMD graphics window that we see when you are striping it, that it actually contains this information. So if you go here down, so you see here. So maybe do a few more here. Go from 12 to, okay, good. So good. And now you see these amino acids appear here. Now you can cut and paste this information out and put it in a command, like in a scripting part, so that you call, this is beta sheet 1. And then you go quickly stripe here, you cut it out, this is beta sheet 2, beta sheet 3. That way you can then color the beta sheets. And so I go here, and you see here is what Fatima had done earlier for us. She colored these beta sheets, beta sheet 1, 2, and 3, with different colors, so that you then recognize which one is which. And then when you are when you're looking, for example, you can do this for trajectory. When you are doing this, you can see then you can now do VMD not only on one data set, but rather on all the frames of a trajectory. And as long as these sheets are still there, they are being colored the same color. And here you see why this is very interesting. Here we pull the ubiquitin between the lysine 48. You see we added here a label. We hold it fixed at lysine 48, and we pull on the terminus. And see, we can stretch it, but we can only stretch the ubiquitin this end. So if you, I don't know, it's a little hard to say because I cannot stop it now, but if I would stop it, you can see you cannot stretch further. This is the shortest, this is the longest connection between that terminus and that lysine. You cannot pull it further. Now, however, if you are going to pull between the two termini, then you can pull it much further. Yeah, so you can see the one time you connect them one way. That also had the tendency that when you now pull on this tag, you can only pull it that far. Whereas if you put them tail to tail, tail to head, tail to head, tail to head, then you can pull it much further. So you see immediately that this connection has actually real relevance for the flexibility of this molecule. Yeah, I mentioned our second pet protein, BPTI. BPTI is actually also an interesting protein. Even though it's so old, it is still not known. And so you can make genuine discoveries with BPTI. Let me quickly tell you about it. So BPTI is a very small protein with only 58 amino acids. It is very rigid. And what it does, it binds to an enzyme, trypsin, to inhibit it. Trypsin is a protein, an enzyme that cleaves proteins. Here you see how it combines itself with a peptide bond, the enzyme and the peptide, forms a complex, and then the protein peptide bonds are cleaved. But when you add to this trypsin BPTI, it's also protein, can also be cleaved and is cleaved actually, but it's cleaved very slowly because BPTI is very rigid. Now why, how, one actually still doesn't know really. And so this is something one still wants to understand, why this particular enzymatic reaction just is arrested and doesn't continue, except it goes very, very slowly. And so in this regard, it's still an interesting protein, but for us it's also wonderful because it's so small and you can even simulate it in a vacuum. It's still very stable and shows some interesting properties. Of course it exists in water or an aqueous environment, but it can be simulated well in vacuum without falling apart. So this is a question why it's cleaved so slowly, and you might want to study this on your own. But you can also already today unpack your pet protein, and after you're through with our examples, you're doing then the real interesting stuff, your protein, and see then how VMD works on this, and you will go and learn a few higher level functions of VMD, and you might actually realize that this gives you some very powerful tool into your hands for your protein, and you might already today realize this is really a very important intellectual tool. I cannot tell you how often we had experimental people, also theoretical people, but particularly experimental people who sit in our room. We have a very nice projection system. We have a big screen so that many people can watch at the same time, but you can also just sit in front of a monitor with your friend and look at the same situation, and then just go with them through the protein, and then they say, oh, I remember this is a conserved residue, let's see what it's doing. Oh my God, it is in such an important place. It holds everything together there. Yes, now I understand why it is conserved, and so on and so on. So really do it and thereby learn it. Even you, all of you must have some protein that they brushed with at one time or other. Take that protein from the PDB, hopefully it has a structure, and look at it with VMD, and you will probably be really excited and love it. And those of you who know already VMD, there was some expert here sitting somewhere, then you know you hopefully learned something a little bit more, and otherwise if you didn't, forgive me, but hopefully tomorrow and the next days you will also be taken care of. Okay, so we have a few minutes for questions. Can be anything. Also, I forgot to give you opportunities for questions after my first round of welcome. If you don't have questions, then I will be an exam. So better ask. What's your favorite protein? RAS. Oh, RAS. Oh, yes. You talk to Jon Kostin. He works on RAS, and you might have something in common there. Yeah, so that's great to look at. You know, like it's a signaling protein that undergoes and has a switch, yeah, it has a switch. Everybody talks about it. If it's trivial what it is, you know, here's a switch, but how it really works, nobody knows. And so, at least with VMD, you can look at it carefully. Sorry, there was a question here, yeah?
My pet isn't that protein at all. It's a silicon structure with alkaline chains on it. Is it also suitable for that?
Yeah. So you will learn to build the files that basically tell tell VMD about the structure of the molecule. So in case of proteins, we have a program that we call PSFGen that generates automatically what is called the protein structure file. But you can take the protein away. It basically tells about the molecular structure. Now, in your case, you would have to build your structure, the topology of this compound into this file, so that then VMD knows where all the bonds are. And so you might just want to try this. There's actually, these PSF files have been actually invented by the CHARM community, as far as I know, widely used by them at least. And for many substances, they have files available, and so you could actually try them. For example, CHARM is also, no, I do marketing, but I market actually here the competitor's product, program. CHARM is being sold through, it's called still MSI or something, through some company. I'm pretty sure they develop PSF files for all kinds of substances, because they are actually more in the market of all kinds of materials and proteins, and so that you might get this from there. We have in the area quite a few knowledgeable people, like in our chemistry department, and so if you have trouble, I would be glad to see if we have a quick fix for you. But it's definitely possible. We use VMD also for gold surfaces, for example, and for other polymers. So the unfortunate thing is with these kind of systems is it reminds me a little bit of my childhood. Like, one of my favorite books was about this little animal that made it into this is German, so I cannot translate. It's a country or a place on Earth where the streets are covered with sweets, and the houses are made of chocolate, and rivers are lemonade and Coke, and everything is fantastic, and the mountains are from cake. But to get into that country, you have to eat yourself through some awful, again, a German dish which every German kid hates. And so huge walls through this, and then you are in this paradise. And so I never knew as a kid. I loved to read on these people who were in this country. You only had to open your mouth and sweets were flying in your mouth. But you had to go through this. And now molecular modeling is like this. The hardest part is to get the structure file, and as you will also learn, to get for particular cases also what is called the force field parameter file, if you want to model now your subject. And so you very often have these sweet students, smart students come, and you have to frustrate them the first half year of their existence out of their wits, and then they have all the fun. So it goes like this, and then it's easy. But there's little one can do about it. Okay. Yes, we also do nucleic acid simulations. We didn't include it in the workshop, but that is something you can do on your own. Yes, definitely. We do. Yeah.
I have a follow-up question. What about, you guys going to be talking about like free energy calculation?
Free energy, yes, we do one free energy calculation, but not with the normal umbrella sampling method, but with our steered molecular dynamics method. But we have a paper out, actually not out in press, where these two methods are compared, and you will be showed there that they are sort of equally complicated. Like typical, mathematics usually doesn't give you any free ride, and so you will do free energy calculations with the steered molecular dynamics method. And what we do is we stretch a helix, and we calculate the free energy to turn the helix into a random coil, and then we let the helix go back to helix and stretch again and from these motions and the work we have to do, we calculate using something called the Jasinski identity, the free energy that is needed to go all along this way. And for that we do But then there's another method called umbrella sampling, where you do not stretch like this, but you go like, you stretch and then you keep fixed, and then analyze the method. You stretch, keep fixed, analyze, stretch, keep fixed, analyze. That's called umbrella sampling, because every time you keep it then fixed, then you actually, the system really goes like Gaussian in a certain umbrella here, like this, and that is sort of like a very equivalent method. So one way you're going to learn. Oh, yes, 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 we do a lot like this. So you will actually do several exercises. Easily, I don't want to say, but you definitely can use it as a molecular editor.
Can you do sequence alignment of
Not yet, but you will do sequence alignment with methodologies that are not integrated in VMD yet. So on Tuesday there is Bioinformatics Day, and but we expect, we hope actually we have a programmer on this, so the ideas are there, and now the programmer is trying to put it into VMD over the summer, and then the problem with VMD is a problem, the strength, and the problem is that VMD is a program for that runs on every platform. So whenever we develop something, it has to work on everything, and that always takes an extra while. I think you want to kick me out here now. Okay, good. So then, do we have now a break or afterwards?
I think the